I thank Ralph and Syed for the opportunity to present Susavion Biosciences today. Our platform takes advantage of the fact that cells of the immune system express an extensive array of regulatory lectin-type receptors on whose ligands are sugar molecules.
809,
so we
500.
have designed peptides as mimetics of these sugars, and we focus particularly on GalNAc and sialic acid galactose, that disaccharide sequence. We have targeted receptors, particularly Siglec-10A, which occurs on macrophages and dendritic cells, and NKG2D, which is on natural killer cells. The way we've designed these peptides, in fact, the one I will say most about today, we discovered by screening a phage display library, and that was our initial approach. We have also used computational, oops, there we are, wrong hand. We've also used in silico molecular modeling to test a fit into the binding site of these lectins, and also then to predict binding energy. From the binding energy, then we design the peptide, synthesize the peptide as quadrivalent or tetravalent structures, and then in fact test the binding in a solid phase binding assay. And what I'll show here in this graph, it shows that if we use a single peptide, a monovalent peptide, we get a very small amount of binding. If we use a dimer, we get increased avidity, and with a tetramer structure as this, we get relatively high avidity to these lectin receptors, and this is a common feature of multivalency to increase avidity. This peptide that I'll be describing today we call SVL4, which is a mimetic of GalNAc, and it binds to the receptor CLEC-10A. The 10A, CLEC-10A, also called CD301, is a receptor that's specific for galactose or N-acetylgalactosamine in mice. There are two separate receptors, one specific for galactose, one specific for N-acetylgalactosamine. But in humans, there's only one, and it's specific for N-acetylgalactosamine. Not much is known about this receptor except that it does contain an ITAM, which is an immunoglobulin tyrosine-based activating motif, which suggests it is an activating receptor. As I said, it occurs on macrophages and dendritic cells, and there's a story behind why we selected this specific receptor, and if you're interested, I can tell you later.
Mic, test two.
No, not working. There we go. With this peptide, it's been injected IV into rats.
No.
It shows actually remarkably long in vivo concentration in the blood, the TK curve that's on the left, showing both the linear as well as the logarithmic plot. And after 90 minutes, there's still a substantial amount of the peptide remaining in the blood. The PK curve is very typical for a peptide injected sub-Q, with a peak at 1 hour and essentially no longer detectable after 4 hours.
Oh, sure.
The peptide is remarkably stable, even at high temperatures, for 14 days. It's stable at pH 3, which is common for peptides, even at high temperatures. It's also stable under oxidative conditions, hydrogen peroxide, 40 degrees for 2 days. And it's relatively stable in rat plasma. At room temperature for 2 days, we recovered 45% of the peptide remaining. In dog plasma, it's even more stable. We recovered 85% of the peptide. We've also looked extensively at toxicity in several studies. There's no evidence of toxicity, whether it's done as a complete clinical chemistry panel or necropsy. There's no evidence of toxicity nor antigenicity. So we tested the biological effect of this peptide with this type of an experiment. We used a mouse model of glioblastoma. And the way the experiment was designed is that glioma cells were implanted into the brain of mice at day 0. At day 7, the mice brains were imaged by MRI to detect whether the tumor was there and to randomize the mice based on size of the tumor. At 15 days, the mice were imaged again to monitor tumor growth. At day 20, blood and tissues were recovered for flow cytometric analysis, and then the mice were allowed to live until they died, another group. So there were 4 groups in the long-lived study. The first group, First group was sham treatment with just injection sub-Q of sodium chloride. The second group was injection of the peptide at 1 nanomole per gram sub-Q on alternate days. Another group received radiation, brief radiation 2 times at day 7 and at day 9, and none after that. And then the fourth group was radiation at 7 and 9 days, and then injection of the peptide 1 nanomole per gram on alternate days as long as the mice would live. This plot shows the survival of these 4 groups. The control group died at 21 days with no other treatment. Peptide alone, interestingly, even though it activated macrophages and dendritic cells, as I'll show, did not really extend life of the mice. Radiation had, in this case, a rather significant effect. This is variable, some experiments less, but significant effect. But when the two were added together, radiation plus peptide, that the survival of the mice were extensively, dramatically increased up to 38 days. Now, we think radiation has an effect on damaging the tumor. We don't think the immune system can really address or attack a solid tumor, as this glioma is, the implanted glioma. But when the tumor cells are damaged, it allows the opportunity for phagocytic cells to attack and clean up the tumor to some extent, as much as it can. During this time, there's a dramatic decrease in the size of the tumor, particularly with peptide plus radiation. The tumor was much smaller than in the other treatments. So we then looked at markers, activation markers, maturation markers on blood monocyte-derived cells. So this shows that the peptide alone, in this case we're looking at the peptide alone, that group of animals, looking at blood macrophages and dendritic cells, that increased these markers, these maturation markers, particularly MHC class II molecules, which are antigen-presenting proteins, CD11B, CD11C, and this is a primary marker for dendritic cells. And then CD115 is just a general monocyte-derived cell population. So you can see there are dramatic increases in the response, in the markers, the extent of markers on the cells in the blood. We then, with this same system, now looking at the group of mice that had both radiation and peptide, and this shows the position of the tumor. And so in these experiments, the tumor-bearing half of the brain was obtained surgically. The cells were dispersed and then analyzed by flow cytometry. We also then, as a control, used the other hemisphere as well to look at what effect the peptide and the tumor had in that side. In the brain, in these monocyte-derived cells in the brain, particularly the brain dendritic cells and microglial cells, looking at the dendritic cells, there's very strong increase in maturation markers, the antigen-presenting MHC2 class, and CD11C. In brain microglial cells, we could also see an increase in these activation markers, indicating that the microglia, being the brain analog of macrophages, the phagocytic cells, that these two cell types apparently were acting in concert with radiation to reduce the size of the tumor. So in this study that we've done, we found that this peptide promotes maturation of blood and brain antigen-presenting cells, as shown by the increase in cell surface markers, activation markers. The peptide extends survival of the mice extensively in combination with low-dose radiation. And again, we think the radiation is simply to damage the tumor cells and make the cells more accessible to phagocytic activity of the immune cells. But also by doing that, these cells present antigens to the immune system and can generate antibody production to really boost the immune system to provide tumor immunity. As I mentioned, we have no toxicity of this peptide as detected in mice, rats, or dogs. And we think this technology could be effective in addition to current standard of care in humans. And in humans, a glioblastoma is removed by surgery, which you can't do in a mouse. And so only small residual remnants of the tumor would be remaining after surgical resection. And we think under these conditions, continued radiation, which is common in treatment of humans, and with the activation of these cells with the peptide, that this could be an addition to a current standard of care. And so we're trying to develop this further. Our company has 12 compounds with demonstrated activity. We have a strong IP portfolio. Our strategy is to focus on infectious diseases, oncology, which I've described today, and also wound healing. The company contains really 3 people. It's David McCallum, our CEO, Dr. Laura Eggink, who is here as our president, and I serve as the CSO. So thank you.
Questions? Gary.
Is the SVL4 injected directly into the tumor?
No, it's always injected subcutaneously somewhere in the body, in the back usually. No, it's not. So it actually probably enters the blood. Some of it must be transported to the brain, but we also think those cells that are activated in the blood also are able to penetrate the brain and attack and provide some of the activated immune cells in the brain.
You showed some PK data. What about the biodistribution data? Because if they're mimicking GalNAc, classes of GalNAc, then there are scavenger receptors and hepatocytes, which for this GalNAc.
I mean, tissue distribution,
yeah.
We haven't looked at that. We haven't looked.
Because you'd lose a lot of your fraction to the liver, I'd imagine.
Sorry,
you'd lose a lot of fraction to the liver.
Possibly do, but we haven't looked at that.
What's the binding affinity of these peptides? Have you measured that to the receptor?
Not directly. Again, most of the binding affinity we looked at, not this one, another one, we've looked at by microcalorimetry, which has given us a nice affinity number, which is in the nanomolar to micromolar range. We have most of the binding studies have been done just with a solid stage binding assay, which requires quite high affinity to survive all the washings that one does in this assay. So from studies with this one, also some of the other peptides that we have worked on, it looks as though the affinity, or perhaps I should say avidity, is in the nanomolar to low micromolar range.
Other questions? Please join me in thanking Ken.